Good morning to the second day of the MLCSB COSY. I have one small organizational announcement. If the third speaker of this first session could please come to the stage and upload his or her talk to the main computer, I would be grateful. Is he or she here already? No? Okay, then we'll have to do it later. But now, let me welcome you again to this second day of the MLCSB COSY. I'm Carsten Borgward from ETH Zurich. I've been the president of the MLCSB COSY since 2015. I'm very happy how MLCSB has developed over these 4 years. Now, as you may have seen, last year and this year, MLCSB was in fact the largest COSY at the ISMB meeting. We didn't anticipate that when we made the plans to create a COSY in 2017. So we were not even among the first generation of COSYs. We joined a bit later than the founder COSYs, and it's all been going very well. And I would like to take the opportunity now to thank all the people who have contributed to this year's success, because in the late afternoon today we'll all be tired and tortured by the heat. So I'll use this time slot now to do this. So a big thanks goes to Manfred Klaasen and Anna Goldenberg, who have been the program chairs this year. You've done an outstanding job and put together a very strong program. And also a big thanks to all the keynote speakers who made this meeting also so attractive, to all the other speakers, to all the reviewers who assessed contributions to MLCSB, and to the entire committee that helps to organize MLCSB and to keep it running over the years. So it's been a pleasure to work with all of you. It's also my last ISMB as the president of MLCSB, so a big thank you to all of you. You are all part and reason for this success that we have seen in MLCSB over the last few years. Thank you very much. One thing that I would like to point out, which Manfred already mentioned yesterday, but I would like to stress it again. So we have a Wikipedia-style homepage for MLCSB. The URL is shown here. And if you go to that website, you see a link to our Google group. And this is essentially a mail list for people who are interested in our community. And you can post anything there, from job offers to events that you are hosting or holding. So feel welcome, feel invited to go to this URL and to sign up for our Google mail list. There are already around 800 researchers working in our field who are members of this group. And if you are not yet, then now is a good chance or a good time to join. I would also like to thank Nico Beerenwinkel, in fact, who jumped in as a session chair yesterday. You heard that Anna Goldenberg had unforeseen personal reasons to not attend. So Nico, who is in fact not a member of the committees of MLCSB, agreed to jump in as a session chair. This was very friendly of him, and I thank him for his support. Okay, wonderful. So we continue with a very exciting program. The first speaker of today is Bradon Carter from MIT, and then I invite him to come to the stage now. He's presenting a proceedings paper entitled DeepLigand, Accurate Prediction of Mhc Class 1 Ligands Using Peptide Embedding. We're looking forward to your talk, and I'll switch the slides. Here we go. And this is... Thanks.
Hi, everyone. Good morning. My name is Brandon, and I'm going to present to you our group's work on DeepLigand, a method for accurate prediction of MHC Class 1 ligands using an unsupervised peptide embedding language model. This is work by Haoyang Zhang and David Gifford, unfortunately both of whom are unable to attend the conference this week, so I'll be presenting on their behalf. The MHC presentation of antigens on the cell surface is a very important part of the immune system. In our cells, proteins are consistently downgraded into peptides by the proteasome. These proteins either could have been internalized from outside of the cell or could have been produced by the cell itself. These peptides can bind to a special class of proteins called the major histocompatibility complex, or the MHC. And once bound, these MHC peptide complexes can be presented out on the cell surface to be recognized by T cells to facilitate a further immune response. This is a very important part of the immune response because it allows the immune system to detect which cells are self and which are infected. In recent years, modeling of peptide MHC binding has been very important, particularly for neoantigen discovery. Neoantigen refers to peptides specific to tumor cells that generally arise due to hypermutations. And some of these mutated peptides are presented on the cell surface. These are so-called neoantigens. And which peptides will be presented, if we can computationally predict that, can be used to help build vaccines against tumor cells, because if we know which peptides are predicted, we can administer them via a vaccine, which will then trigger a strong immune response against the tumor cells with hopefully a high degree of specificity. I'll talk a little bit more about the MHC Class 1 pathway. So there are multiple properties that must be satisfied along the way in order to MHC to present a particular antigen on the cell surface. So first, when the protein is degraded into peptides, there must be certain motifs present in the protein to allow proper cleavage. Additionally, there needs to be an efficient transport of the peptides into the ER by a molecule known as TAP. And finally, there needs to be a high degree of binding affinity between the MHC allele and the peptide itself. I'll also point out here that everyone's MHCs are just slightly different to allow each individual immune system to identify self antigens, and so it's important that there's a high degree of specificity in order for the MHC peptide complex to bind and then make its way up to the cell surface where the peptide is presented. Most existing work thus far has focused on computational modeling of this aspect here, MHC peptide binding affinity, largely due in part to the large data sets that are starting to accumulate in databases such as the IEDB containing this kind of data. However, because these models only tend to take into consideration this feature, they often overlook these other pathways which in our work we attempt to model as well. To talk a bit more about the type of data that we're using, recently mass spectrometry has been used to identify these natural ligands. We can engineer one or multiple MHCs on a cell surface, allow them to present natural ligands. We can pull down the MHC peptide complex using immunopurification, elute the peptides from the complex, and then use mass spec analysis to identify the amino acid sequences of those peptides that are the natural ligands of those MHCs. So the existing methods that then use this data include MHC Flurry and NetMHC-Pan 4.0. I'll describe both of these. In MHC Flurry, as we said before, this is an affinity model that is a neural network that attempts to predict, given a peptide and some MHC, the binding affinity between the two. NetMHC-Pan 4.0 is very similar, although it also contains an additional output node, which is a classification of whether that ligand is eluted or not. And here, because the features, the earlier layers of the neural network are shared, there's the strong assumption that the two tasks here share the same set of features. In our approach, DeepLigand, we predict whether a peptide is a ligand or not based on two independent modules. So the first is the traditional MHC peptide binding affinity, but in addition, we also have sequence features of some of the other auxiliary factors in ligand determination. So as I said before, this was the TAP efficiency and also proteasome cleavage. And here we have an unsupervised module that we train that hopefully captures those and then is combined with the binding affinity network in order to predict ligand or non-ligand for the peptide and MHC. One question here is, how do we actually model these auxiliary features since we don't have a proper data set for them? All we have is the result of the mass spec analysis, which were positive sequences that actually were ligands. We don't know anything about the peptides that didn't survive the process. So our approach for this is to consider a language modeling task of the peptide sequences. So I'll turn to an example of natural language processing where language modeling is very common. So in that application, generally we have corpora of, say, English text from sites like Wikipedia or newswires, and we never saw anything that was sort of wrong or negative sequences. All we had were valid English sentences, and we trained an unsupervised model. So in that case, you might say, given all of the words you've seen thus far in the sequence, predict what the next word is. And so we didn't need any particular label. We can still learn things about the English language, about syntax, grammar, and semantics, having only seen the positive sequences. And we hope to apply a similar methodology here to the peptide sequences. So we train a language model on the natural ligands, and this language model then embeds them as a peptide embedding, which is a high-dimensional latent representation of the peptide sequence that hopefully captures something about the semantics of important parts of the MHC peptide presentation process. For a language model, we use the recently published ELMO model, which learns a conceptualized word embedding. So to talk about the specifics of ELMO, we have 3 layers. There's a local convolutional layer, and then 2 stacked bidirectional LSTM layers, one of which runs in the forward direction, one of which runs in the reverse. The forward and backward direction of each of these 3 layers are concatenated here. And in the original ELMO work, these embeddings produce, so for a given token, we can find the final embedding as a linear combination of these 3 embeddings, where the weights of that linear combination are learned with the model. In our work here, for simplicity, we simply take an average of those 3, and so use this approach to, for each peptide in our peptide sequence, produce a high-dimensional embedding. The key here for ELMO, that it makes this a very powerful model, is that unlike simple language models where a single token is always embedded by the same vector, here the word embedding is dependent on the context around the word. Because the embedding is a function of the LSTM, which is a function of the entire input sequence, both in forward and reverse, the particular embedding for each token is a function of the context around it. And this is a very powerful and rich representation that hopefully captures information. Next, I'll talk about the other component of DeepLigand, which is the more traditional binding affinity prediction. For this, we'd use a deep residual network. So the inputs to this network are a peptide sequence and an MHC. The MHC is represented through a pseudosequence, which represents the residues of the MHC that are closest in 3D proximity to the peptide, and so are responsible for binding. The 2 are encoded, their amino acids are encoded using a concatenation of a one-hot embedding and also a BLOSUM 50 representation, which captures similarities between amino acids and is commonly used in these types of models. These encodings are then combined and are the input to the deep residual network, whose output is then combined with the peptide length in 2 final fully connected layers. And eventually we predict the mean and variance of a Gaussian affinity distribution. From this distribution, the mean gives you an idea of a point estimate or the expected value of the binding affinity as predicted by the model, and the variance allows us to first model any experimental noise that was present in the data collection, but additionally gives us a measure of model confidence or uncertainty in the prediction. So as the variance is higher, we can interpret that as the model being less confident in that particular decision. So then these 2 models are trained independently and then combined through a final layer to predict ligand or non-ligand. Compared to previous models, so as we said, there was MHC Flurry and Net MHC Pen 4.0 are 2 common existing approaches. DeepLigand outperforms these approaches as measured by both AUROC and AUPRC. However, we note here that other methods can be trained on other data sets that we don't have access to, and so their approaches, so what we do is we adopt their specific approaches, implement them in our particular architecture and our data sets, and then evaluate them. So I'll talk about that next. So first we have an approach we call affinity only. This is akin to the strategy employed by MHC Flurry, where we only train an affinity-based model. In this case, then there's no unsupervised language model, no peptide embedding, but we take the affinity model to be our deep ResNet. So it's sort of the same approach we're using in DeepLigand. Our second strategy is two-task, which is akin to the strategy employed by NetMHC Pan 4.0. And here we take our deep ResNet and add the additional output node for whether the ligand is eluded or not. And so again, now we're forcing the model to share features for both tasks. Finally, we evaluate whether we can discriminate ligands based on our embedding-only model. So this is now the output of only the unsupervised language model, and add it through one fully connected layer to make the prediction of ligand or non-ligand. And I'll note here that there's now no knowledge of the MHC representation in this strategy. We're purely using a representation of the peptide to make the prediction. Again, we see here now with our same architectures and on our same training data, we again see that DeepLigand performs as alternative strategies as both measured by ROC and PRC. And one interesting point here that I'll note is that the embedding-only model, which is shown in red, actually performs quite well. And this is interesting because, as I said, there is no knowledge of the MHC, and this is implying that the peptide embedding is capturing meaningful predictive information in order to discriminate ligands from non-ligands. So our next question is to actually explore what those features might be and to interpret the embedding-only model to understand what features it's capturing. To do this, we adopt the recently published sufficient input subsets technique, which is an interpretability method for interpreting pre-trained black box models. Here, the interpretation is given by a minimal set of the input features whose values alone allow the model to reach the same decision as on the original input to a particular level of confidence. So the idea is that we take a sparse subset of input features, mask everything out, and the model can arrive at the same decision. And then we can interpret that subset of features as the rationale or the basis for the model's decision on that particular example. Here we're interpreting the decisions for a large number of peptide sequences that through the embedding-only strategy were predicted to be natural ligands. They all have, in this case, length 9, and then we cluster those into 6 different clusters and show each cluster through a sequence logo. We can compare these logos to a known motif of proteasome cleavage, shown on the right here, and we see there's actually very nice alignment between the two. So just as one example, if we look at position 9 in some of these clusters on the bottom here, we see there's enrichment for L, Y, and F, which if we look on the motif on the right, we see again L, Y, and F are high information content containing residues at position 9. Upside down characters shouldn't be there. And so we see there's nice alignment between the two, which is suggesting that our unsupervised language model is capturing information, meaningful information about earlier parts of the peptide presentation pathway, in this case, cleavage motifs by the proteasome. Finally, we're seeing here that DeepLigand is able to classify ligands from non-ligands, even among sequences with medium or weaker binding affinity. So looking at the plot on the right here, we're looking at AUROC performance as a function of the range of predicted affinity. So the units here are in nanomolar, so smaller means stronger binder. So on the left here, we're seeing that in the affinity-only model for weaker binders with medium binding affinity, the performance of the model is actually barely better than random guessing. While in contrast, DeepLigand is achieving very high classification accuracy regardless of the range. This is important for applications such as neoantigen discovery that we talked about earlier, because neoantigens tend to be binders with medium affinity, since they're both non-self and also tend to be rarer. And so it's important that for that application, our models are able to computationally predict accurately both for medium and for strong binders. So in summary, we see that DeepLigand, by combining a traditional binding affinity model with an unsupervised peptide embedding language model, outperforms state-of-the-art approaches at predicting MHC Class 1 ligands. We see here that then when we adopt the strategy of combining both the unsupervised language model with the, in our case, ResNet, it is superior performance to using either of those approaches individually. We next explore the features learned by the unsupervised peptide embedding language model, and we show that in this case they are highly predictive of ligand identity, and we also see that they capture known motifs of proteasome cleavage, as we hoped the language model would learn something about earlier parts of the pathway, given just positive peptide sequences. And then finally, we see that DeepLigand is able to accurately predict even when the predicted binding affinity is weaker and we're not predicting just the strongest binders, which is important for applications such as new antigen discovery. We have an implementation of DeepLigand available on GitHub. And as I said, this is work by Haoyang Zhang and David Gifford. I'm happy to try to take any questions you may have, but otherwise you're also very welcome to send them any further questions directly via email. Thank you very much.
Thank you. Any questions? I think Manfred is on his way. Yes.
No, I made it to the microphone. So, if I understood correctly, the language model you learn for the peptide sequences only. And I was wondering whether you also considered to extend this language model to also include the protein sequence context of the peptide in order to possibly get better embeddings that are more informative.
Yeah, so you're correct that these are just right now on the positive peptide sequences. I think that that'd certainly be an interesting area for future work. I'm not sure exactly what you'd expect to happen, but I think it'd certainly be an interesting area to explore, definitely.
And then, like a smaller technical question, is it overkill to use a deep residual network for the affinity module? Did you also try out less complex residual networks and assess performance?
Yes, I'm not sure how shallow they were, although I know that there was some various tuning of the model architectures, and this model was identified to be best through cross-validation evaluation. So I know that various architectures were tried, so this ended up performing best.
Okay, thanks.
Hi, thank you for the talk. So for the MHC complex, are you including just the sequence of the residues that bind to the peptides in your model, or are you also including the three-dimensional positions of these residues?
It's just the former. So the MHC is represented by a pseudosequence, which are, I believe, the 34 closest residues to the peptide in 3D space, but we're not actually encoding information with the 3D structure. It's just based on that pseudosequence, which is, in our case, effectively one-dimensional, and then we embed that with the amino acid encoding of a 1-hot and the Blossom 50.
Okay, thank you.
Thanks.
We have time for one more question. Yes, please.
Yeah. Hello, thank you. I was wondering how you could extend this kind of method to just common proteins, so with a few thousands of sequences in an alignment, and if there would be scaling problems with long sequences of 200 amino acids, for instance.
Yeah, I think that would certainly be interesting. I'm not sure in terms of scaling. I mean, these architectures should hopefully be, certainly able to perform inference on sequences of that length, I think. And so I think that would certainly be an interesting area for future work. Okay. Thanks.
Thank you. If there are no more questions, then let's thank the speaker again. Thank you.
Thanks.
And we have one